Welcome, AP Biology, as we start to talk about Chapter 4, A Tour of the Cell. Um, so, we're starting to get into some areas that I think you're probably going to be more familiar with, and some terminology you probably remember from your middle school and junior level bio classes. Now, we're going to dive a little bit deeper into how this cell works. So, the first distinction when we're talking about cells that I want to bring up is the difference between prokaryotic cells and eukaryotic cells. So, these are going to be things like bacteria. Our prokaryotes are going to be things like bacteria and archaea, and then everything else on Earth, so plants, animals, protists, fungi, those are all going to be eukaryotic cells. And we can see that the prokaryotic cells definitely look a lot simpler, whereas the eukaryotic cells have much more compartmentalization and some organelles. And that eukaryotic means a true kernel or that center nucleus. So Dora here will tell us the differences between eukaryotes and prokaryotes. D for DNA. In prokaryotes, the DNA is circular and it's naked. It's just out in the open. Whereas in eukaryotes, it's bound by proteins, it's linear instead of circular, and it usually has introns, which are these non-coding regions that break up genes that'll be excised when we're reading the DNA. O for organelles. The prokaryotes have no nucleus, no membrane-bound organelles, and 70S, which just means smaller ribosomes. When we compare that to our eukaryotes, we have a nucleus present, that true kernel. We have membrane-bound organelles, things like mitochondria and chloroplasts and vacuoles, and then 80S ribosomes, which means they're a little bit bigger. Reproduction is going to be a little bit different. Prokaryotes do something called binary fission, whereas in eukaryotes, there's either mitosis for most cells or meiosis for sex cells. And then in eukaryotes, there's multiple chromosomes, whereas in prokaryotes, there's just that single chromosome. And then finally, prokaryotes are typically a lot smaller on average than those eukaryotes. So those are some of those differences between prokaryotic cells and eukaryotic cells when you hear me talk about them. So we're going to dive into prokaryotes a little bit later. We're really going to be focusing on eukaryotic cells. And I want you thinking, what benefit would membrane-bound organelles provide for a cell? So why would it be beneficial for evolution to give these cells organelles? Well, why are we going to do organelles? Well, it allows for specialization, so we can have different tasks being done in different parts of the cell. In prokaryotes, it's just kind of all over the place. Membranes that divide the cell allow for different environmental conditions. So when we specialize and compartmentalize, we can have different pHs, different concentrations of molecules. So if you're trying to do a very specific job and you need a lot of calcium, these membranes let us keep all our calcium in one place. Or if you're breaking down things and you want it to be really acidic, we're going to keep those acids in one place so that they're not destroying other parts of the cell. They provide a surface for reactions. When we get into respiration and photosynthesis, you're going to see that the interactions that play from moving molecules back and forth across a membrane end up providing these huge chemical reactions that give us all the energy to do everything we do. And then specialization is going to be a prerequisite for multicellular life. So why do you think we would want specialization if we're going to go to multicellular life? Well, if we think about our bodies and we have all these different organ systems that do different jobs, it's a very similar concept to those organelles where they're doing different jobs. And so different cells in our body are going to need more or less of certain organelles. And so not all your cells in your body look exactly the same. So we're going to really focus on plant cells versus animal cells. Now you may want to say like, well, Mr. Malgren, I'm a mycologist and I love fungi. And so why can't we talk about fungal cells? They're closer to plant cells. And yes, there's going to be some differences. Like instead of using cellulose, they use chitin, but we're not going to worry too much about that. So plant cells are going to be photoautotrophic, which autotroph means they provide food for themselves. Photo, light, they're using light to photosynthesize using those chloroplasts. Plant cells are going to have cell walls made out of cellulose, which we talked about before was a carbohydrate, a long chain carbohydrate that is good for structural integrity in those cell walls. And then plants have a large central vacuole that they're going to fill up with water, and that's going to help them maintain their pressure. If you start to lose water in a plant cell, the membrane will peel away from that cell wall. And that's what it's going to look like if you don't water a plant, or if you buy some lettuce and it gets too dry and it starts to wilt, but you give it some water, the vacuole fills up, it pushes against that cell wall, and it becomes firm again. Animal cells, on the other hand, are heterotrophic. They have to eat other stuff. They can't make their own food. There's no cell wall, so they're going to be a little bit more gelatinous, and they have discrete, smaller vacuoles. So instead of having one big place for everything, they're going to have small ones distributed all throughout the cell. And there's going to be advantages and disadvantages to both of these adaptations. So what makes life life? So what makes a living thing? Now, it may be easy to look at a rock and say, like, well, that's not alive. And then look at a tree and say, well, that's alive. But when we start zooming in, what actually makes life life? What are those criteria for life? Well, something needs to be able to respond to its environment, so respond to stimuli. It needs to be able to grow and change. It needs to be able to reproduce. It has to make more of itself. It needs to maintain homeostasis or balance. So it has to be able to regulate itself in order to stave off entropy. It needs to have some sort of metabolism, so processing of energy. And then it needs to have some sort of hereditary way to pass along its traits to offspring. And so that's going to be kind of part of that reproducing. And then there should be some form of movement. And so when you think about things like water may be able to do a lot of these things, but it can't do all of these things. Maybe rocks can do some of these things, but it can't do all of these things. And unless it can do everything on this list, we don't consider it alive. So let's start going through what makes a cell a cell. And every single cell is going to have a plasma membrane. Now, this is going to be a phospholipid bilayer with embedded proteins. We're going to get more into the plasma membrane in our next lecture because it is so cool. But for right now, you need to know that it has these little phospholipids, which means lipids that have a phosphate head on them. And they are amphipathic, which means that part of it is polar, part is non-polar. And so those hydrophobic tails will form the inside of the layer, and the hydrophilic heads will be on the outside. And then there's some embedded proteins on the surface or on the inside, or channel proteins that make holes in it so things can move through. And then you'll also find that there's some carbohydrates and cholesterols and other lipids that are embedded into this that we refer to as a fluid mosaic. Now, what's its whole purpose? It's kind of like the exterior gate of the cell. It's going to control what comes in and out of the cell. And then it also is in charge of communication, so sending and receiving messages from outside of the cell, so communicating with other cells or the environment around it. And so, as I said earlier, eukaryotes are bigger than prokaryotes, but they're both pretty small, ranging from 5 to 50 micrometers. And if you don't know what a micrometer is, well, there's 10,000 micrometers in 1 centimeter. So we're talking really, 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 really small stuff here. And so why can't cells get bigger than that? Why can't there be a car-sized cell that's moving down the street? What might prevent a cell from growing in size? What problems could be faced with our car-sized cell? Well, let's think about how things move around in a cell. Most particles are going to move around by diffusion. So diffusion is spreading from a high concentration to a low concentration. So we can kind of see that random movement in these GIFs here of particles where the doors open, where we have a high concentration, and they're going to start to spread to that low concentration. So diffusion isn't going to be a lightning quick reaction. And if you think about something like traffic where you have to merge in, entry of particles into the cell is going to be limited by the amount of membrane space available. So if the membrane is in charge of keeping things in and out, if you have more membrane, there's more ports of entry, there's more protein channels, so more stuff can get in. And then if you have stuff coming in from more places, you can fill the cell with more nutrients faster. But as you get less membrane, there's going to be less opportunity to fill the inside with all those nutrients that you need. And so, well, but you may say, Mr. Mellgren, I'm pretty good at math, and I know that if you get bigger, there's going to be more on the outside. Well, that's true. But unfortunately, math will also tell us that surface area does not grow as fast as volume. So for pretty much all shapes, volume is going to increase more rapidly than the surface area. So we can see that here, where in this cube here, if every side has a length of 2 centimeters, our surface area is 24 centimeters, and our volume is 8. Remember, you just add up all the sides, the area of all the sides to get 24 square centimeters, and the volume 2 times 2 times 2 is going to give us 8. So that gives us a surface area to volume ratio of 3. Now, if we go up to sides length 4, and we do 4 times 4 times 6, it's going to get us 96, whereas 4 times 4 times 4 is going to get us 64. So now that ratio is 1.5. If we go up to again to 6, 6 times 6 times 6 is going to give us 216. 6 times 6 times 6 is going to give us 216. Now we're exactly the same. So you can see, again, here, if we go up to 8, now our surface area to volume ratio, our volume is bigger than our surface area. So you can see, as you grow in size, volume is going to go a lot bigger than surface area. Now remember, volume is everything that's inside, everything that needs to be supported by those nutrients. Surface area is your ports of entry. So even though surface area is getting bigger, it's increasing at a slower rate than what you have inside. And so if you want to grow as a cell, it makes more sense to just make more cells than to get bigger. And this is going to be the big evolutionary step for why single-celled life grew into multicellular life. Because cells hit a ceiling of how big they could get. And if they wanted to keep growing to be the dominant species in the ocean, then they had to get clever and start working together to increase in size. And so you can see here, the same volume, if we have it in one cell versus 27 small cubes, we have the same volume, but the surface area here is 5,400 micrometers, whereas the surface area here is 16,000 micrometers. So a big difference in that ratio. And that's going to allow those nutrients to get in and diffuse through the cell much more efficiently and keep that cell alive. So let's start going through some of these organelles we talked about. Let's start off with the big brain in the center, that true kernel, our nucleus. Our nucleus is the home to the genetic information. It's frequently referred to as the leader of the cell or the ruler of the cell. And that's because all the information for what that cell needs to do and what it's going to make is encoded in there. There is a double membrane around the nucleus. And inside of the nucleus, at its very core, you will find the nucleolus, where all that DNA is being stored. Now, this is the site of DNA storage and replication, but it's also going to be the site of where mRNA is made, that is shipped out to protein or to ribosomes to make proteins. So, since I just mentioned proteins, it's probably a good time to think back to last chapter and think about what they do. Proteins do everything. They build molecules, they digest molecules, they carry out chemical reactions, they provide structure, they copy our DNA and RNA, they receive and send messages to the environment and to other cells. They are regulating everything that's happening inside those cells. So they're super important. And from our nucleus here, the DNA is transcribed into mRNA. That mRNA, for messenger RNA, is sent out to our ribosomes where it's read, and amino acids are put together through our good old friend dehydration synthesis until we have a polypeptide chain which gets folded up into a protein that goes and does a job. So proteins are going to be the most important thing we have in our body because they're doing everything. And so, like I said, those are made at the ribosome. Now, ribosomes are a complex of both RNA, so it's part nucleic acid, part protein. And there's two subunits, as you can see, a large subunit and a small subunit. And those are going to come together like a hamburger bun on top of that mRNA that it reads to make proteins. As I mentioned earlier, those eukaryotic ones are a little bit bigger, and they are protein synthesis. They're going to use RNA to read that mRNA, and they're going to start attaching amino acids together to make a long polypeptide chain, fold it up, make a protein, and you have the recipe for making everything that makes the cell run. So where are you going to find those ribosomes? Two places. There's free ribosomes, which are just floating in the cytoplasm. And I realize I haven't said what the cytoplasm is. So your cells have to maintain that shape, and they're filled with a medium, mostly water, but it's a goo, the jello that's inside of us all, cytoplasm, the medium that fills the cell, basically. So there's some ribosomes floating around, and these are going to be for making proteins that stay in the cytoplasm. So anything that's staying inside the cell, doing work inside the cell, those are going to be on free ribosomes. Ribosomes can also attach themselves to something called the endoplasmic reticulum. And these are going to be bound ribosomes, and they are going to make proteins that go to the cell membrane and also make proteins that may get shipped to other cells. Now, ribosomes are not locked into their job. They can switch from becoming free or bound ribosomes based on the RNA that they're reading. So if I'm floating for free in the cytoplasm and I grab some RNA that needs to be shipped out, it'll bind itself to the endoplasmic reticulum. So since we're talking about the endoplasmic reticulum already with ribosomes, let's get into what the ER is and what it does. So you have two types of endoplasmic reticulum. The rough ER, which is covered in ribosomes, which is why it's called rough. It looks like those candy bit paper with candy dots on them. And then the smooth ER, which is a little bit further away from the nucleus and has no ribosomes. So it's going to look like a smooth series of tubes and pipes. Now, the function of both of these is to move biomolecules through the cell and process biomolecules, but they're going to work on different things. So The rough ER is going to be for protein synthesis. That's why it has all those ribosomes there. So the ribosome is going to build the polypeptide chain. That is going to move into the ER, and it's going to get processed, folded, and then sorted and sent where it's supposed to go. The smooth ER is going to synthesize lipids. It is also going to detoxify certain toxic compounds. It's going to add a hydroxyl group, which you'll remember is an OH, which is going to allow it to be dissolvable a little bit easier in water, which is going to help it flush from the system. It's also going to be involved in breaking down glycogen, which is long-term energy storage. And it also holds a lot of calcium, which is really important in things like muscle cells, for muscle contraction. So smooth ER is going to be really big in your muscle cells. Additionally, when things are shipped out of the ER, they're going to end up at something called the Golgi apparatus, also known as the Golgi body, or you may even hear of it as the Golgi complex. And this is going to be a series of flattened membrane-bound sacs. So what do I mean by that? These are kind of gelatinous, almost look like pancakes, and they are going to be fusion of different membrane vesicles that form into these larger structures that sort and organize where things are supposed to go. And then it's going to modify, mark where those biomolecules are supposed to go, and then ship them off in new vesicles to send them where they need to go in the cell. Now, I just mentioned the word vesicle. What is a vesicle? A vesicle is a small little compartment surrounded by a membrane. They're sort of like little cell bubbles, and their function is going to depend on their contents, but usually it's to carry things around the cell, or to store things in the cell, or to separate or compartmentalize in the cell. Vesicles are made by budding off or fusing into membranes. So like, as you can see in the picture here, the external environment, some molecules come in and that membrane will close around them to form a little vesicle that will then safely transport those molecules somewhere else. Now, all these things I just described are part of the endomembrane system. Now, the endomembrane system is really important to understand how it works. So let's track the journey of a protein that's traveling to the outside or exterior of a cell. So the RNA is going to travel to the ribosomes on the outside of the rough ER. And then that's going to build a polypeptide chain inside of the rough ER, where there will be certain proteins inside that will help it fold into the protein it needs to be. And then it is going to move through the ER, sort of like a highway, until it gets to a place where it can be packaged into a little vesicle. That vesicle is going to carry those proteins to the Golgi complex, where it is going to move through the Golgi body or Golgi apparatus. And during this time, it is going to get modified a little bit more, and it's going to get tagged for its destination. The Golgi body is going to sort those proteins on where they're supposed to go and clump likewise proteins together and put them into a new vesicle that will then move to the cell membrane and fuse into the cell membrane to either put that protein there or send it into the outside of the cell to go where it's supposed to go. So let's see a little video that shows how this works.
It is important that translated proteins are delivered to their specific cellular location. To accomplish this, the protein is transferred through a series of membrane structures. A principal membrane component is the Golgi apparatus. The Golgi apparatus is the sorting organelle of the cell. Proteins from the rough endoplasmic reticulum are sent to the Golgi. As the proteins move through the Golgi apparatus, They are modified and packaged into vesicles. Because the Golgi apparatus receives proteins from one location and targets them for delivery to a second location, it is sometimes considered the post office of the cell. The Golgi apparatus consists of general components. The cisterna nearest the endoplasmic reticulum, or ER, the medial and transcisternae, and the trans-Golgi network. Other key players in this process are the proteins being transported and the enzymes that modify them. Translated proteins are encapsulated in vesicles in the ER. A group of these vesicles fuse, and these fused vesicles form the cisterna. As the protein moves through the stack, it is modified by resident Golgi enzymes at specific locations in the apparatus. These modifications are important because they provide the signal that determines the final destination of the protein. So how does the protein move through the Golgi? Movement occurs in waves. First, the cisterna becomes part of the medial Golgi cisternae. Behind it, a new cisterna is formed by the fusion of vesicles from the ER. Meanwhile, one of the medial cisternae migrates and becomes the new transcisterna. Collectively, this process is known as the cis-maturation model. Proteins are sorted within the trans-Golgi network. Proteins with the same target sequence are destined for delivery to the same location. The trans-Golgi network then buds off into vesicles. These vesicles then migrate to their target location. These locations include internal organelles, such as the lysosome, the digestive organelle of the cell. The vesicles can also be targeted to the cell membrane, where the targeted protein can be released from the cell for delivery elsewhere in the organism.
Cool. So then let's also look at the journey of a lipid. So lipids are synthesized in the smooth endoplasmic reticulum. Lipids that are headed to the membrane will be packaged in vesicles and sent to the membrane. And lipids headed to other organelles, like the mitochondria, will be grabbed by transporter proteins, which will grab onto them and shuttle them to their final destination. So a little bit simpler system there. Next up, the cytoskeleton. So much like your skeleton, it's a big component of cell structure, and new microscopic techniques have allowed us to fully realize the intricate network of these filaments in our cytoskeleton, as you can see in these pictures here. And there's a few different components. Microtubules, which you can see here and here, are hollow rods made out of proteins that support the cell structure. Remember, if you don't have a cell wall like in an animal cell, this is going to be extremely important. And they also provide tracks for motor proteins to walk along and carry things throughout the cell. There's a centrosome, which is a region within the cell where all microtubules grow out of and are built from. And at the center, there is a pair of centrioles, which are extremely important for cell division. And then there's microfilaments, which are things that you can see here and here. A common one is called actin, which is really important for cell contraction for things like muscle movement. And then finally, intermediate filaments, which play a huge role in anchoring and structure, making sure things like the nucleus and organelles stay put, and they don't all just sort of sink to the bottom of the cytoplasm. So cytoskeleton, big in structure and also in movement. So what do those motor proteins look like? Well, let's take a look. Here we can see a microtubule that a motor protein is moving along, and carrying a vesicle behind it. And this may look silly, but this is what's actually going on. And the chemical interactions between the heads of these motor proteins and the microtubule are causing that movement where it binds to the next level, binds to the next level, and then they'll also bind to things like vacuoles and be able to move those storage vacuoles around the cell. Pretty cool. The lysosome. Lysosome is one of my favorite organelles, and honestly, you should remember what the lysosome does because it's one of the College Board's favorite organelles. And it is a little sac, a little vesicle filled with hydrolysis. We're seeing those same words come back. So breaking down digestive enzymes. And so what does it do? Well, the lysosome will fuse to vesicles or vacuoles that have nutrients, and so it'll help break down large molecules into smaller components. It will digest damaged cell parts. So if something's not working quite right, it will add its digestive enzymes and chemicals and help break down those damaged cell parts. And then it's also involved in apoptosis, which is cell death, which is programmed cell death, the destruction of the cell overall. And so when you're thinking about lysosomes, it's important to know that the cytosol or the cytoplasm has a pH of 7, so a very neutral. But the lysosomes maintain an internal pH of 5, which is acidic. And so why would that be? Well, this goes back to why do we have organelles? If we can keep all the acidity in one place, then it's really easy to break things down because you have that acidic environment to help those digestive enzymes. If you don't keep it very acidic and it's just spread out throughout the cell, it's really hard to focus those enzymes and focus those acidic properties on what you're trying to break down. And since we're talking about apoptosis, I always think this is super cool. You were actually made by apoptosis. So way, way back when you were just growing in the womb, your hands looked like these frog flippers. In fact, your fingers, your nostrils, and any other gap or hole in your body grew as a result of apoptosis, where you had a big old flipper, and then apoptosis broke out your fingers, making those space for your digits. Pretty neat. Lysosomes are also very involved in diseases. In fact, there's 40 known lysosomal diseases that are often fatal. And you can imagine what would happen if your digestive enzymes stopped working. If your lysosomes grow but can't digest and disrupt those cellular activities. One such example is Tay-Sachs disease, a genetic disease where because they're not working, undigested fats build up in the brain, and that causes severe developmental issues and typically death by age 4. So it's very sad, but tied to a single organelle not working. Next up, we have vacuoles. Now, vacuoles are just big membranes, big giant vesicles filled with storage materials. So maybe it's food vacuoles, maybe it's water that you have in there. There's also something called a contractile vacuole, which you'll find in cells that live in water. These pump excess water out of the cell. So these vacuoles will fill up with water, and then they use those microfilaments of the cytoskeleton that we talked about to pull on it, compress or contract that vacuole, and force water out of the cell. Pretty cool. Very cool. Mitochondria. So mitochondria, which you probably all know, is the powerhouse of the cell. Well, that's true, but it's important to know what's going on and how that works. So the mitochondria is going to be one of our more complex organelles in terms of structure. It has an outer membrane as well as an inner membrane that's very folded to give it a lot of surface area. So within that inner membrane, there's the cristae, which are those folds of the inner membrane, and then the matrix, which is the fluid-filled cavity inside of the inner membrane. Now, if you look at that, it says that it contains ribosomes, enzymes, and its own unique DNA. What? Well, once upon a time, and we'll talk about this more in the future, the mitochondria was a prokaryote that got swallowed by a bigger cell, and instead of digesting it, that small mitochondria was better at making energy, so it kept it. And the mitochondria loved this deal because it didn't have to worry about protecting itself from being eaten anymore. It was safe. And so then as that cell, that one action, divided, that ended up becoming all eukaryotic life stems from that one cell that ate that mitochondria. Really cool stuff. So what's its job? Well, aerobic, which means oxygen, cellular respiration. So making ATP, or the energy currency of the cell. And so it's just cranking out energy for our body to use, cranking out that energy that we use. What you'll find in plants, and we'll talk about later in more detail when we talk about photosynthesis, is chloroplasts. Now, these also have a double membrane and also have their own DNA because, shocker, this was also swallowed up by a cell that became the foundation of all plants. And inside, you'll find something called the stroma, which is the liquid inside inner membrane, the cytosol of the chloroplast. And you'll also find thylakoids, which are stacks inside the inner membrane, and that's where ATP is made, and that's where photosynthesis is going to happen. So what happens in chloroplasts? They turn light into sugar, and so we'll be talking about that later when we get to chapter 8, I believe it is. And the mitochondria and chloroplasts kind of go back and forth. Mitochondria take sugar and oxygen and make it into ATP energy, carbon dioxide, and water. And then chloroplasts take carbon dioxide and water with some energy from sunlight and make sugar and oxygen. So it's kind of a reverse process going back and forth, back and forth, back and forth. Now, big time misconception alert that I want to clear up. Only plant cells have chloroplasts, only animal cells have mitochondria. Wrong. Animal cells do not have chloroplasts. That is true. But plant cells have both mitochondria and chloroplasts. Plant cells still need to turn that sugar into ATP, which it can readily use. ATP is the energy we use. Sugar is a storage molecule. So that's why we have both of them. And so chloroplasts are manufacturing sugars that mitochondria will convert to ATP. So if we want to get some of that sweet, sweet sugar, we got to eat it. Whereas plants, they can just make it. It's pretty cool. So what are the big takeaways you should leave this lecture with? You should have a basic understanding of the function of each organelle. You don't need to be an expert on each organelle, but you should have a pretty good understanding of like a one sentence, what does it do? You should be able to describe in detail, though, the endomembrane system. So how do proteins go from the nucleus to the outside of the cell? Talk about those steps along the way with the vesicles, the Golgi, and the endoplasmic reticulum. You should be able to know some of the differences between eukaryotes and prokaryotes, as well as between animal cells and plant cells. You don't need to be experts, but you should be able to rattle off a couple details. You should be able to describe why cells have an upper limit on their size. So you should be able to, in your explanation, relate that to nutrient usage and nutrient diffusion throughout the cell. And then you should be able to explain the evolutionary origins and purpose of organelles. We'll get into that a little bit more in depth with those evolutionary origins and swallowing up those smaller organelles. But you should be able to describe some of those things about compartmentalization and why some of those functions of each organelle help get to multicellular life. If you can do those things, you're looking pretty good. So hopefully you'll ask me any questions if you have them. But that was our tour of the cell. Hope you enjoyed.